Woodland, not an open savanna setting.
Now that's a surprise.
These are woodland animals.
That runs counter to so much of what
Runs counter
people
all the way
expect.
back to the 1800s.
For over 100 years, there's been one main theory of how our ancestors started to walk on two legs. The theory goes like this. We started out as apes that lived in the trees. We looked like chimpanzees and walked on all fours using the knuckles of our hands. But the climate changed. And only after forest had turned to savanna did we start walking on two legs instead of four. Artie tells a very different story. She was already walking upright while living in the woodlands. Artie's skeleton ran counter to the old theory too. It was unlike anything anyone had seen before. It took a team of experts 15 years to fully piece her together. So how do we know she was bipedal? The man in charge of figuring out how Artie might have moved was anatomist Owen Lovejoy.
The critical thing in bipedality is the structure of the pelvis. And at first, when you look at this bone, you might think that there's not much information here. But actually, there's an enormous amount of information that tells you a great deal about what the original bone looked like.
Using information from this deformed bone, the team was able to reconstruct Artie's entire pelvis.
This is a plastic structure that's produced by a three-dimensional printer.
The top part of Artie's pelvis looks more human. The hip bone is short and broad, a key indicator of bipedalism. But the lower half is much longer than a human pelvis, more useful for climbing.
When you got to that detail, you realized it was a mosaic of anatomy that had never been encountered before.
It suggests that Artie could walk on two legs, but not as well as later hominids like Lucy. And the rest of her anatomy held surprises too.
Neither Lucy nor humans have the ability to grasp with their big toe, but this thing had full grasping ability.
So she has a grasping foot that can walk.
Yes.
Even the hands were a mosaic. Incredibly long fingers, good for climbing, but a shorter human-like palm. And the bones show none of the telltale signs that she walked on her knuckles like a chimpanzee. So we now have a detailed picture of the crucial time when our ancestors had just started walking on two legs. Artie was small, around 4 feet tall. A good climber, she moved on all fours when in the trees, but she walked upright when on the ground. She blows the old theory of how we became bipedal out of the water. Not only did our ancestors start walking on two legs when still living in the woods, they never looked or moved like our closest living relative, the chimpanzee. Some scientists don't accept this interpretation. They think Artie isn't a human ancestor at all, but the relic of an extinct ape. But Timms spent years studying many specimens of Ardipithecus, and he thinks the evidence is clear.
When we looked at the Artie cranium, we saw very small canines. No other ape, living or fossil, has such reduced canines. That's a good indicator. She's come in our direction a bit. Go to the pelvis. Same thing. A unique adaptation shared by humans and Lucy.
We're still not sure exactly why a woodland climber needed to walk on two legs. But thanks to Artie, we now have a snapshot of one of the most dramatic transformations in the history of our species. The transition from walking on four legs to walking on two. This major change to our ancestral body plan has serious consequences for us today. These consequences can be seen within our own bodies, and they're not all good. This vault in the bowels of the Cleveland Museum of Natural History is the place to go if you want to find out what can go wrong with the human skeleton. Dr. Bruce Latimer is an anthropologist and anatomist who was a curator here for many years.
Well, this is the Hammond Todd collection, which is the largest collection of its type in the world. There are over 3,000 skeletons in this room. In each one of these drawers is a complete human skeleton.
They come from unclaimed bodies at a Cleveland morgue dating from the early 1900s. If you study these skeletons, one thing becomes clear pretty quickly. The human back goes wrong. A lot.
There's an enormous amount of back problems. The pain must have been phenomenal, and you just see that all over this collection.
Turns out, our bad backs are an unwelcome inheritance from our inner monkey.
We took a skeleton like this that was essentially horizontal. We stood it upright. We've had to change essentially every bone to allow us to do that. And we forced it into this new position. We have a problem going from this kind of animal into that kind of animal, and our main problem is balance.
On all fours, the weight of our body hangs down from our spine. But turn this body upright, and the weight is all out front. It puts us out of whack, totally out of balance.
In order to balance, we had to create this curve on your back. And then your head would be back here, wouldn't it? So we had to create another curve here in the middle of your chest, and then your head would be sticking out in the front, so we had to create another curve in your neck. And so we have this S-shaped curve in our spine, and we expect that to hold us up. That is an engineering nightmare.
Our S-shaped spine is unique among mammals, and it causes all sorts of problems. The vertebrae that make up the bones of your back and the discs between them are put under a lot of pressure, particularly the ones at the very apex of the biggest curve, the thoracic vertebrae.
If you take those thoracic vertebrae and you push on them from the ends too hard, what happens is this. You end up with what's called a wedge fracture. That curve has crunched it. No other animal has anything even remotely like this. It's a consequence of how we walk.
And it's not just fractured vertebrae. From slip discs to sciatica, our spines go wrong in all kinds of ways. 80% of all Americans will complain of back problems at some point in their lives. Our inner monkey has a lot to answer for. But I don't want you to get the wrong idea. Standing up on two legs wasn't all bad, or it never would have happened in the first place. It freed our hands. An anatomical change that, combined with our amazing brains, would eventually allow us to make tools and reshape the world around us. This key moment in our evolutionary history is once again visible in the rocks of northern Ethiopia.
So geologically, one of the things that's special about this is it has lots of layers. Lucy's from one part of it, and we're going to another. Right?
That's right. We've just jumped about 600,000 or 700,000 years in time.
And that is kind of mind-blowing.
Yeah.
Paleontologist Bill Kimball has brought me to a site where you can find stone tools that are over 2 million years old, if you know what you're looking for.
So what do I look for?
Well, you'll look for flakes on the ground.
Oh, is that bone?
That is a piece of fossil bone, and here's another
See,
one.
I can't find tools like if I'm bone. Yeah. Right. The important thing about tools is it's the hand and the brain, right? It's both together.
Absolutely.
You got one?
Oh, yeah.
Well, I mean,
You got another.
this is very typically
A nice one? I don't get this at all.
Yeah, I know. It looks like something from your driveway.
The tool Don's found was made by human ancestors called Homo habilis, who lived 1 million years later than Lucy.
Clearly, some early human took a stone, a hammer stone, and struck it right there. And got
Oh, yeah, you got striations,
You have to feel that edge. You could use it to strip off meat or whatever.
So from this rock, you can tell where they hit it, how they hit it. I mean,
Yep.
it's almost like a time machine you have.
It is. It's a time machine that takes us back to a period when the faintest glimmers of what it means to be human are beginning to emerge. The use of the hand, the cognition, the repeated behavior to produce a stylized tool.
It's mind-blowing.
Wow. Can we look for tools? I want to find a tool. No? That's natural.
No, this is one. You found one.
Making tools takes excellent vision, fine motor skills, and a brain that can integrate the two. We humans only have this sort of brain because it's been shaped by our ancient primate past. Monkey, ape, or human, our brains share a basic architecture that's different from most other mammals. We all have a special region involved with hand-eye coordination, and a greatly expanded visual system with as much as 40% of the brain involved in seeing. Our inner monkey hasn't just shaped our bodies, it's shaped our brains as well. But if you look at the modern world around us, and everything humans have been able to build, from satellites to skyscrapers, there's no doubt we're more intelligent than monkeys. So what changed? What made our brains so different from those of other primates?
Hi, Neil Shubin.
Hi, Neil. It's Tom Verbacher.
Good to see
Welcome
you.
to the Infant Primate Lab.
So this is the monkey place,
This is the monkey place.
Tom Verbacher studies monkeys in order to answer questions about our own brains. He's going to show me an experiment that demonstrates an important difference between our brains and the brains of a monkey. But the differences aren't what you might expect, especially when we're young.
Monkey on the set.
Monkey on the set. Neil, this is C.I. She's a 3-month-old pigtail macaque.
This experiment tests something called object permanence. In other words, whether this monkey knows an object still exists when she can't see it. It's a developmental milestone for both monkeys and humans.
You're going to put a piece of fruit on the toy itself. You'll place it in the well,
She's looking at it. There.
and you'll move that to completely hide that toy.
Okay.
Oh. There you go.
Wow.
There you go.
Wow. That is impressive.
Object permanence.
3 months old, and already C.I. understands object permanence.
C.I., you are good. Hi, Geneva. How are you? You are a cutie.
Geneva is the same age as the baby monkey. I'm going to do the exact same test on her under the expert eye of child development specialist, Professor Susan Speaker.
She needs to reach for him and show that she's interested.
Oh, I think we got interest.
She's got a good reach.
You do. Look at you. Oh, Geneva, you like those. Hello.
Now, Neil, can you get them away?
Oh, I'm an expert at this. Can I take that for a sec? Sorry. Be temporary. Yeah, you are so good that way. I'll pop it in.
Make sure she's watching.
She's watching. Now, we'll cover it up. Watch this.
And she wonders, what did you do, Neil?
What toys?
I don't know where they went.
Wow, that is amazing.
Where did they go?
It is incredible. If it's not there, it doesn't exist. That is amazing.
So how come a baby monkey beats a baby human in this test?
These tests are showing us how rapidly a particular region of our brain is wiring up in development. And it's showing us comparatively that we wire up much slower in this part of the brain than do monkeys.
Compared to other primates, we humans have an unusually long childhood, even if you take the different lifespans into account. This should be an evolutionary disadvantage. But in fact, it's one of our greatest strengths. During childhood, our brains are being shaped by our environment and by our experience. Extending this phase gives us longer to learn and pick up new skills. Some think that this is a key part of what makes us so smart. But there are other factors too. The brain is made up of vast networks of nerve cells called neurons that carry, process, and store information. The part of the human brain responsible for higher thought, the cortex, contains more neurons than that of any other animal on the planet, an astonishing 16 billion. These neurons are organized in a uniquely powerful way. They're unusually interconnected. This means we can do things that other primates can't: build complex tools, compose symphonies, and even investigate how our own brains work. But before we get too carried away with our own cleverness, I want to inject a bit of humility. That means leaving monkeys behind and going back to where the series began, my anatomy lab in Chicago, and my favorite subject, fish. The brain of a fish and the brain of a human have more in common than you might think. This is a trip to the beginning. This is as basic as it gets. We're going human fish, represented by a cartilaginous fish, a shark on the right. And what you have is, you see the muscles and nerves and organs inside the shark head. But importantly, you see this yellow tissue. This is the front end of the spinal cord. It's the brain. Doesn't look like much, but it's the shark brain. This is the human brain. This is where it all happens for us, where our memories reside, where our fears, our loves, our hates. It's all in here. The human brain, really at the surface level, looks nothing like what's sitting inside the shark head. Yet when you know how to look, you find a very profound similarity. And that similarity lies in the overall architecture of the brain itself. In the human, we have a forebrain. And that forebrain consists of this folded tissue here, where much of our conscious thought happens. And then there's a midbrain, this walnut-sized area in the middle. And then a hindbrain. That's a fundamental division. We see that in development, and we see that based on where the nerves exit the brain. Well, if you follow the similar nerves in a shark, what you see is sharks too have that same structure. A forebrain, a midbrain, and a hindbrain. There's a fundamental architectural similarity among all brains. The brains of people, the brains of dogs, the brains of cats, the brains of monkeys, lizards, frogs, salamanders, trout, bass, even sharks and fish are similar. Each one of them has a forebrain and a midbrain and a hindbrain, despite the fact that those brains are often very different in function and form. But that's not the end of the story. The fundamental architecture of our own brains can be traced back even further than fish, to a surprisingly primitive animal that doesn't even really have a brain. But I've got to find these tiny creatures first.
Right, okay. Well, we could start trying around here.
X marks the spot?
X marks the spot. Let's give it a go.
This is Peter Holland, a world-renowned geneticist and head of zoology at Oxford University in England.
Okay, so what do I do?
Just shake it.
Shake and bake, huh?
I'll make it look so easy.
That? No?
No. They'll be pretty obvious because they'll really swim around. The water makes the digging slightly harder.
It does.
You got one. There's an amphioxus.
We got any? Whoa, look at that little guy go. Hey!
Just dip it in the water again, you'll see it'll flick around.
Well, they really flick.
There you see. There he is.
Hey, it's beautiful. Look at that. Which is the front?
It doesn't look like much, but this tiny creature, called amphioxus, has much to tell us about our own brains.
That is cool. Okay, that ranks as cool.
Okay, so let's take a look at a couple of these.
So I brought this from the anatomy lab in Chicago. This is a human brain. When I look at this, I don't find any obvious similarities to that.
Deep in the genes of this animal and the development of this animal, and deep in the genes of us and the development of us, there are the similarities. They're pretty active. They're pretty active.
Holy cow. Oh, that is beautiful. God, they're so clear, too. You can see right through them. You know, it's amazing.
Amphioxus lives in the sand of the ocean floor, filtering algae out of the water. It's from an incredibly ancient group of animals. we found fossils that look a lot like this in rocks that are over 500 million years old. They are a window into what came before the first fish. And crucially, they don't have anything like what we'd call a brain. The simple nerve cord that runs the length of their body ends with just a tiny swelling invisible to the naked eye.
If you look at the front end of this animal, you don't see it all expanded into a large skull or brain region. It's just pointed.
But if you look at the genetic makeup of these ancient creatures, you find something truly remarkable. To make the forebrain, midbrain, and hindbrain that we saw in our lab, you need a series of control genes to tell the cells within the developing brain where they are. They're like a zip code. Those same control genes that make our own brains are active in amphioxus. But here, they're simply building the primitive nerve cord and the tiny swelling at its front. This means that the genetic roots of our complex primate brains existed in much simpler creatures that first arose over 500 million years ago.
I think it gives us a glimpse into where our brains came from, into the basic organization of the brain of our ancestors.
I mean, I find that mind-blowing.
It's just an exciting animal.
He knows you're talking about him. He's going.
That's right. He's going. You can see those muscle blocks really working there. Yeah, and I think to an evolutionary... Well, he's jumped off, guys. Sorry.
He's going TikTok on us. He's going land-living. He's gone road.
Yep, he's gone for the next transition.
Exactly. He's in the Devonian now.
The search for our inner fish has taken me from high in the Arctic.
Here was the snout of exactly the creature we were looking for.
To the plains of Africa.
Yeah, I just want to run down here and start collecting fossils.
Let's go.
As well as deep inside our own DNA.
And today we saw a little tiny worm living in the mud here in the bay that contains genes that we have that sculpt our own brains. The organ that gives us many of our unique properties.
We've seen how our distant relatives, from worms to fish, from reptiles to early primates, have defined our bodies. At each new stage of our journey along the tree of life, our ancient animal ancestors have reconfigured what's gone before. And that's how we ended up as the intelligent creatures with all our quirky flaws that dominate the planet today.
There's something incredibly profound, and I think beautiful, in the idea that inside every organ, cell, and gene of our body lie deep connections to the rest of life on our planet.
Your Inner Fish is brought to you in part by viewers like you. Thank you. To learn more about this program, visit pbs.org/innerfish. The series Your Inner Fish is available on DVD. The book Your Inner Fish is also available. To order, visit shoppbs.org or call 1-800-PLAY-PBS.